This is a production of Cornell University.
Hey, thanks for the introduction, Tony. So I'm really glad to be here today. This is my first time talking at SIPS in general since my job talk 2 years ago. And so I'm excited to tell you about some of the things that I've been working on since then. So I thought I'd start by giving you a little bit of a professional introduction to who I am, since I haven't met many of you before. So I am a plant population and community ecologist by training, and my dissertation was on range expansion of plants in response to climate change. When I did that work, I developed a couple of research themes, including creating models of ecological processes for individual plants, and then scaling those models to populations and landscapes. Did this though, as a graduate student, I eventually realized that I didn't want to just be creating this knowledge. I also wanted to be applying it. And so since then, I've been working on another research theme, which includes using these models. If I can get the pointer right up here, using these types of models to optimize health-related ecosystem services and disservices. This has brought me from the remote forests of northern Michigan to more urban landscapes. And I've also turned my focus more from plant demography to plant-human interactions. For example, shade. So right now in Austin, it is where this photo is from. It is right now today, it is 92 degrees there. Honestly, a little jealous at the moment, but it is, so in that context, shade matters an awful lot. So we have trees like this one providing shade. And now let's zoom out and look at that same tree from above. And here it is. And what we can do is we can model these ecosystem services like shade. And for example, we can include things like the structure of the trees, like their three-dimensional shapes, as well as the trajectory of the sun across the sky to model something like shade over the course of a day. So here's an example of a little animation of shade. Now, because this is built with remote sensing data, we can then scale this up to neighborhood or municipal scales. These models help us to capture how these different ecosystem services and disservices vary. And when it comes to something like shade, we have variation from one species to another. For example, a big oak tree is going to produce way more shade than a little crepe myrtle tree. But there's also these important differences between species based on their biology. So for example, live oak keeps its leaves throughout the winter, whereas shimard oak is deciduous. And so we need to understand these little pieces of the biology in order to create good models, and in turn, leverage this information to do a better job of optimizing things like shade. And so this is the type of question that I look at. Things like, can we use these differences in shade provisioning from one type of tree to another in order to do things like keep people out of the emergency department during heat waves? So I'm starting up a project right now on this exact question, and in it, we are doing things like quantifying where trees are throughout New York City, as well as how different types of trees provide cooling, either through shade or evapotranspiration, things like that. We are then plugging that information into municipal scale climate models, and then taking that to understand heat exposure for people and assessing how that heat exposure results in emergency department visits through epidemiological analysis. And ultimately, with that type of information, we can create things like risk maps of where somebody is most likely to end up going to the hospital for a heat-related morbidity. And since all of this is integrated throughout, we have the potential to play around here and see what happens if we were to have more trees of this type in one neighborhood or more of them in a different type in that neighborhood. And so ultimately, this gives us a way to understand what's going on and to see what will happen if we pull different policy levers, like planting more of one type of tree versus another. So this is a project that I'm working on with a couple of collaborators, and I'd just like to take a moment to thank Arnab and Chi, who are over in Weill Cornell and engineering respectively. And with these kinds of projects, they're just way too big to deal with it all on your own. So it's really emphasizes the importance of interdisciplinary approaches and collaboration. What I'm going to do now is shift over to a different type of interaction that we have with plants, and that is through airborne pollen. Now, almost all gymnosperms and about 10% of angiosperms are wind pollinated. So they release their pollen into the air and hope that it gets to the next plant. This is important for all sorts of things, whether you're talking about crop pollination or the spread of herbicide resistance genes and weeds. But today I'm going to be focusing in on the public health relevant aspect of this, which is through allergies. So I got really interested in this topic after grad school and ended up doing a postdoc at the School of Public Health at the University of Michigan, where I developed a municipal scale model of airborne pollen. And I approached this by doing things like figuring out how much pollen a plant produces as a function of its size. So a lot of allometric equation development, a lot of also mapping of these plants with remote sensing and field studies, as well as developing phenological models. And then I threw all of these together to get things like this, which are estimates of pollen release on particular days based on the individual trees, how much pollen they're creating, and when they're releasing it. And so this model here shows you pollen release over the course of a season. And these are for oak trees. I suppose this one is the spring of 2017. And so you can see this variation over both space and time when it comes to pollen release. This also is important to airborne pollen concentrations. It's sometimes assumed that just because pollen can blow long distances, that it will. But it turns out, based on this work, that where there's a lot more pollen production, shown on the x-axis here, there's a lot more pollen in the air on the y-axis. And this is intuitive. It's hardly surprising that there's more pollen near the plants that are releasing it. But it's also really important because it gives us a way of understanding the marginal effect of a particular plant on something like pollen exposure. But this was just a model for an individual city. And plus, I already told at least some of you all about this project. So I want to move on and scale this up. And so after I finished that project, I moved out to Texas, specifically to the University of Texas at Austin, where I did another postdoc working with Elizabeth Matsui and Shalini Jha. There I focused in on Juniperus ashei, which is sometimes called mountain cedar, blueberry juniper. It has, I think, about 6 common names or so. But what sticks with it is not so much any of those names as the fact that it is a prodigious producer of allergenic pollen. In fact, the locals in central Texas call it cedar fever when they're exposed to its pollen because it produces so many allergic symptoms, things like sinus pressure, itchy eyes. This thing is probably the most hated species in Texas, no small feat. It is also abundant in that region. Here's a map of its distribution there. And the darker red colors indicate where it's the highest density. In many of these places, it's the most common tree species. And this whole area is also, it's home to about 14 million people or so. I think that would make it the 5th largest state in the US, just this one area here. It houses San Antonio, Austin, the Dallas-Fort Worth metro areas. And so there are a lot of people which are affected by pollen from this tree. This photo on your left shows just one little twig, and all of those are individual pollen cones. And a big Juniperus ashei tree can produce literally millions of these things. And inside each one of these pollen cones, there can be as many as about 500,000 pollen grains. Just to give you some perspective, for somebody who's really sensitized to this stuff, it can take as few as like 15 or 20 pollen grains per cubic meter of air to begin an allergic reaction. So you can imagine the suffering that this causes. Understandably, people care an awful lot about knowing when this is going to be in the air and where. And so there's a whole network of pollen monitoring stations. Here are some that are in this area, but the pollen monitoring network run by the National Allergy Bureau has about 80 pollen monitoring stations in the US. The closest one to us here in Ithaca is in Rochester. So let's zoom in on one of those, and I'll tell you a little bit about how it works. So this is Austin, and then so this is downtown Austin here, and then about 25 miles away in Georgetown is the nearest pollen monitoring station. Up on their roof, they have a sampler that looks something like this. And a few times a week, somebody goes up to that rooftop, takes out a slide or the adhesive tape, counts all of the pollen grains on there, manually identifies them, and then reports that concentration to the media. However, that information that they report is often, it's in the best case over the previous 24 hours, and it's also from one single rooftop that's pretty far away from where most people live. And so what does this mean if you live in, say, East Austin? It means that you may not have the information that you need to make good decisions. It may mean that you don't have the information to decide whether you should go for a run that day, or shut your windows, or turn on your air filters, things like that. And it also means that you don't have the guidance necessary to decide whether you should take your medication ahead of time, because a lot of these require 24 to 48 hours to reach full efficacy. So a lot of people have asked me, how good are the commercial pollen forecasts, right? Because you can just open up any of the weather apps on your phone and it'll give you a forecast. It'll tell you what's going on right where you are, right at that moment. And so I really was wondering about that too. And so this past summer and now this fall, I've been working with a couple of folks, including Stark, who's in the audience there. And we've been trying to do this systematically. And so we're developing a approach to pulling in all of this information from all these different websites using their APIs, scraping their websites, all of that. So, and then comparing that to the empirical pollen measurements. We're still in we haven't launched quite yet, hopefully within the next couple of weeks, but we did a little bit of manual curation of some of this data. And I just want to show you that as just like a little teaser. So in this figure here, the date is on the x-axis and the pollen index is on the y-axis. And each of these lines are one of the different commercial pollen forecast providers. You'll notice that they go all over the place. Now I want to show you this line here, which is the actual data. And even though this is pretty preliminary, I think it gives you a pretty good sense of why we need to do better. So what we have now is this sparse network of monitoring stations. I'd argue that what we need is a different system that is both geographically comprehensive, that is transparent and accurate, and also gives us a way to incorporate the underlying biology into these predictions so we can produce good near-term and long-term forecasts. With that type of information, we could help people avoid pollen hotspots. We could tell them when to take their medication. And we'd also be able to get a better sense of the overall public health impacts of pollen exposure. So the goal of my postdoc was to create these models of airborne pollen concentrations for this species of juniper, and then to both assess its effects on public health and ultimately to figure out what we can do to make this situation a little bit better. There's a bunch of different pieces of this project, including things like figuring out where these trees are, how much pollen are they releasing, when are they releasing it. There's a ton of different pieces of it. I'm just going to tell you about a couple of them today, though. And first, I'm just going to convince you that this is important from a health perspective to begin with. And so I wanted to do just kind of a baseline epidemiological analysis to see if we use traditional methods, what pops up. And so what I did was to compare pollen concentrations from those monitoring stations with some health outcomes data. These figures here show airborne pollen concentrations over time. And our study period was about 5 years, from 2016 to the end of 2020. And this first panel here shows the concentrations of Juniperus ashei, which includes the juniper species that I work with here. The thick black line is the average across all the stations, and the thin gray lines are the individual stations. And so you can see that there's a lot of variation. Also note, this is a log 10 scale. I then compared this with emergency department visits where asthma was the primary diagnosis. And I did this for 3 different age groups: young children, school-aged children, and adults. And so basically, I'm going to just skip to the chase here and show you some of the results. On the x-axis here is the date ranging from January to December. The red lines indicate the number of observed cases. And then each of these colors here represents attributable risk. The ones that I want you to focus on are not the viruses, although those are very important, but instead the pollen. And you can see these big spikes in emergency department visits at certain times of year for tree pollen in the yellow, and then also for also some sizable spikes for Juniperus ashei, right around the new year. And so so even with these kind of coarse exposure metrics, we do see some strong effects of this tree on emergency department visits for asthma. I'd also like to take a moment to thank the folks who have been going out and collecting all of this pollen data. Like, this type of work isn't possible without them. And so I'm very thankful that they've allowed their years and years of pollen collection data to be used for analyses like these. Now I want to turn back to phenology, and that is one of the really big questions here. Like, if we're going to create these complicated models, we need to know when the pollen is being released. And to approach this problem, I ended up doing a large observational study where I spent a couple of field seasons driving all around Texas. These blue symbols are places that I visited multiple times, and the hollow symbols are sites that I visited once per season. when I visited these sites, I did things like collect twig samples and count the number of pollen cones that had opened, and then within these pollen cones, the number of pollen sacs that had opened up. And so, let's see. So here is an example of a single tree that was visited multiple times. On the x-axis is the date, and on the y-axis is the proportion of pollen sacs that had opened. And so you can see in early December, nothing had opened up. Then around early January, it really starts to open on up until mid-January. This tree has basically released all of its pollen. So this gets us a certain amount of the way there. It tells us more or less when this particular tree is releasing pollen, like generally so. And so this is useful, but I wanted to take this a little bit further than just linear interpolation. And so I modeled the opening of individual trees using a Juniperus function, which is in some ways has a fairly similar shape to a logistic curve. One of the nice things about taking this modeling approach is that it allows you to both apply it across a lot of different trees within a site, and then also to multiple different sites. And I did this within a Bayesian framework, which allowed me to account for missing data in some places. And there's a bunch of really cool statistics that are involved in all of this. And I have like 12 slides at the end if you really want to get into it. But for now, I'm just going to say some cool Bayesian stats if you're interested. And with this information, we can do things like say what the season midpoint is for a site like this one, or for some other site. In this case, this is one of those sites where it was just visited a single time in the season. When we display those season midpoints geographically, you can begin to see some patterns here, where the season midpoint was a little bit earlier for sites over in, say, the eastern range of the species compared to the western range of the species. And so I was, of course, curious about whether I could predict this. And to do that, I turned to a bunch of different environmental data sets, one of which was SMAP, the soil moisture active passive sensor. And with this, I was able to begin to try and figure out what could predict the timing of pollen cone opening for the species. Now, one of the problems here is this is releasing pollen in the middle of the winter, so we can't use any of those traditional frameworks like growing degree days. They just don't really work. And we don't have very good a priori hypotheses about what periods are important to the development and timing of release for this plant. And so I turned to, in this case, to a statistical approach using the Klimwin package and R, and basically allowed the data to speak for themselves to figure out the periods of time that have the greatest influence on this phenological outcome. And this is what this shows. It basically gives allows us to explore how much of the variation we can predict essentially when we have an opening window, when we open our phenological window of impact, let's say in springtime versus in the fall. And ultimately, we can use this to figure out the predictors that have the most effect here. And those turned out to be soil moisture, which is not a surprise. This is a pretty water limited system. And so these maps show soil moisture from SMAP, both in the first year and in the second year. And then we can compare it with the timing of those seasonal midpoints for those sites. And you can see there's a fairly decent relationship here. The sites which are wetter tend to be releasing pollen earlier. And then what we can do based on those relationships is see how that varies. We can map that back over into space. And here is that same information in an animation. And so the colors here indicate the amount of cones opening on a particular day. Each of these frames is one day. Starting at the end of December, we are now into early January, and you can see this wave of cones opening up, going from the eastern part of the state over to the western part of the state. Of course, these seasonal patterns are important, but if we're talking about atmospheric dispersion models, we can't be content with just what's happening in a given week. We need to understand what's happening on these finer timescales, the ones that are relevant to, say, like a gust of wind coming through. And so I simultaneously investigated some of this stuff on finer timescales. And to do that, I both took a large number of observations myself and also started up a citizen science project. And I'm not going to focus too much on the citizen science data today, but I can tell you that after driving thousands of kilometers, it's pretty cool to have people just like going out regularly, taking some of this data, which is so useful and important. Now, one of the things that we measured was just qualitatively how much pollen came off one of these trees while it was being observed, with a very technical approach of shaking the branch and seeing, was there a lot of pollen? This is an example of a lot of pollen. I've had 4 different people tell me stories about how newcomers to Texas have seen the pollen being released from these trees and thought there was a fire. And like, so the fire departments get regular calls at this time of year being like, something's on fire in the woods. But okay, so what we can then do is model pollen release, whether there's a lot of pollen being released or just a tiny bit, as a function of things like the developmental curve of those trees from before. And we can also include finer grained environmental data, things like wind speed and humidity, vapor pressure deficit, stuff like that. This is an example from GridMet here, which is a gridded source of meteorological data. And this is just, each of these different frames here is just, let's see, this is vapor pressure deficit from one day to another day in early January. So you can see there's huge, the weather changes. It's not consistent one day to another, especially in Texas, or I suppose here. And with that type of information, we can begin to see some patterns. So I'm just going to show you the results for vapor pressure deficit, which you can, if you're not familiar with it, you can think of it as how much drier the air is than a plant and how much water is being sucked out of that plant potentially. And then we can, so let me orient you to some of the results for vapor pressure deficit. On the x-axis here is vapor pressure deficit. And this is on the current day here. So on the day of observation. And then these colors indicate the likelihood of there being a lot of pollen released, essentially. This is relative risk. This is using a distributed lag framework, which is a pretty cool statistical approach, which lets you both have some flexibility in the period of lag as well as the shape of its effect. And it's used in epidemiological analyses, but it applies pretty well to this system too. So what we see here is when it's drier, there's a lot more pollen being released. But we can also do that going back in time too. And so this is the current day. And then up here was a week ago, and up here 2 weeks ago. And we can see that when it was much drier, like a week or two ago, at that particular site, it's much more likely to be releasing a lot of pollen now. And so this gives us a way to kind of untangle some of the relationships. And ultimately, we can use this to predict pollen release over time. So here, this is just at one particular site. Date is on the x-axis and the probability of high pollen release is on the y-axis. And you can see that there's a lot of variation from one day to another based on these environmental variables, as well as on the different ripening rates of these different trees. And just to compare this to those kind of smooth seasonal trends, it turns out that these don't work quite as well for actually getting at what's going on. So including this more fine-grained data was really useful. That being said, there's a ton of limitations to this. Like, for example, I only went out at night occasionally. And so almost all of these observations are during daylight, more or less. And they're also just a few snapshots of this process. And so what I wanted to do was to get at the full pollen release and temporal distribution for individual trees. And so I wanted observations around the clock. And I also didn't want to have to collect that data myself. And so what I ended up doing was to build a device to do this for me. And so I want to introduce you to the pollen platter, which is something that I built with the help of Seth down at UT Austin. The way this platter works is it's a box that you can place underneath the tree. There's a little slit in the box, and then some of the pollen falls through that slit. Underneath the slit is a platter, which is controlled by a little microprocessor and just kind of, and is programmed to turn at a predictable rate. So you can then see the areas where there's a lot of pollen on that platter and map that back over time. And so I just deployed these pollen platters bunch of times, bunch of sites, few different treatments. And and I'd also like to take a moment to thank Kent for all of his help processing these samples from Texas. So here is, I think, perhaps the first comprehensive record of pollen release on these types of timescales from a wind pollinated plant, which I think is pretty cool. And so here we have the date on the x-axis and the pollen concentration on the y-axis. And you can see that there is a ton of variation, both between days, which we kind of already knew, but also over these fairly short timescales, which means that we can't just ignore what's happening on the scale of hours. We need to take that into account. And we can use the same types of approaches as before. We can plug in different environmental data and use that to understand the effects of things like temperature and precipitation, vapor pressure deficit, to create these models on these fine temporal scales. One of the issues that we had with this, though, is that we're kind of merging together all of these different processes, right? There's the presentation of pollen, but then there's also wind, right? The wind isn't always blowing, so it doesn't always fall off immediately. And I thought, God, it sure would be nice if we could just like reduce wind as a variable. And so Kent and I spent a while this summer iterating through different setups here and eventually got something going in the growth chamber where we have a fan on a timer to blow it. And then we can have all of this in a growth chamber so we can alter the things like temperature and humidity to begin to do a better job of figuring out exactly which of these processes matters when. I also, the pollen platter is great, but they can be a little bit finicky, and it's not always feasible to place these out. And so I also worked with Blue, who's an undergrad here, to see if we could do time-lapse photography to capture this process of anthesis. And so these are some examples. This is Plantago lanceolata, and you can just see these anthers popping out over time. And Blue also built his own little setup for manipulating temperature and relative humidity. And so we can begin to tease some of these things apart, and how if we change humidity in one way, how does that affect the rate of anthesis, shown here as both the size of the dot and the color. And we can also see like what happens when we change temperature, when we change both at the same time, and we can begin to really understand what's going on here to a greater degree. And ultimately, the goal with all of this is to merge these different models together, to include these seasonal variables, and also to throw in what's happening on these daily and hourly scales. So I was really hoping that I'd have a little bit of this further along, so I could show you an animation on these hourly timescales using some gridded environmental data, but check with me again in a few weeks. So here, these are just a few different pieces of this process for understanding the pollen concentrations over time and space and their health effects. And oh, I suppose I also wanted to mention pollen production, which is an important piece of this puzzle, and also one which I do not have a good handle on yet. And the reason I'm giving this talk here today is I've been working with a prospective PhD student who is going to be applying to the soil and crop sciences section, to the graduate field. And Hannah is going to be working on figuring out how much pollen some of these trees produce. And we're hoping to make use of the really stark differences in color that you see between the male trees of the species and the female trees. On the left here, you can see a male tree that's this kind of brown color. And on the right, you can see a female tree without any pollen cones, which is green. And so we're hoping to use these differences in reflectance as a way of kind of scaling up pollen production for these trees. So we can, and we're hoping to do this with some aerial imagery, but we're also exploring a couple of other possibilities. Unfortunately, this projector is not really showing the reds very well. So you're going to have to more or less take my word for some of these trees looking much more reddish brown than others. This is supposed to convince you that it's very feasible to do this because there are these big differences, but you will have to take my word for it, I'm afraid. And so once we can scale up things like pollen production over these large areas, we're hoping to compare them to other environmental data, the usual climatic stuff, but also things like soil nutrients as well. And the spin we're putting on this for Hannah's GRFP application is all about the climate change and the synchronicity of plants, kind of going into some of the hypotheses around masting that were developed for trees. So there's a lot of basic research questions, which I think can be answered with these methods too, which are pretty cool, but I'm not going to get into them in too much detail now. There's also some possibilities here for extracting phenological information from trees using high temporal satellite imagery, specifically from Planet, which I think would be really cool and super useful. So I'm investigating that possibility right now. So there's a lot of steps to be done here, but there's also a ton of potential for connecting these basic ecological, geographic, and phenological processes through to their specific effects on human health, and ultimately to figuring out how we can get this information into the hands of people so they can do something about it, whether it's changing their behavior, managing their medication better, or whatnot. And each of these different steps along the way gets us a little bit closer to that goal. Of course, that's just one species and one state. And so ideally, we'd be thinking about how to do this on a much broader level. And so I've been thinking a lot about how do we scale up these types of things to a continental level. And one of the big answers to that question involves collaboration with citizen science, in particular with the National Phenology Network. And yeah, so that is something that I'm actively working on. And it turns out that creating phenological models is something that you can do with enough observations, enough environmental data. You can create these models, and it turns out that they do a pretty good job of predicting airborne pollen concentrations. I just heard back that this little paper here just has some minor revisions. But that is some of where this type of thing is going in the future, scaling up to bigger scales so we can make a difference in more people's lives. So with that, I'd like to thank some of the various funding sources that have funded both my postdocs. And I'd also like to thank some of the folks who have been involved in this throughout the years, especially here in my lab now to Kent, Lou, Stark, and Kyle, as well as to some collaborators elsewhere. And with that, I'd be happy to take some questions. Thank you.
In terms of how after that you will be modeling, so I was wondering if there's a way to make this kind of data collection automatic with the machine vision or other.
Yeah, totally. So there are some groups working on automating pollen identification. There's some nice systems over in Europe that only cost about 600,000 US right now. The exchange rate's good at the moment. But those systems are very expensive, and that makes it hard to kind of scale up with them. There's also a company here in the US, Pollen Sense, that I've been working with a little bit, and they are trying to do exactly that, using real-time data collection, microscopy, computer vision, and so on. And so this is an approach which, even if it hasn't hit its full stride yet, I think will be very useful in the future. That being said, when concentrations vary by orders of magnitude across like a kilometer, you're never going to be able to get the density that you would need to provide good information. And it's also for individual people. And so I think it's going to play a really large role in this, to have this more real-time data collection. At the same time, if we approach this with models, we'll be able to generalize better, and if we understand the underlying biology, we're going to be able to predict things like there not being much pollen release on January 9th and 10th, things like that.
So from what you've shown, I'm getting the sense that the flower induction happens much earlier, and then perhaps those flowers are just sort of quiescent. And then when the conditions are right, perhaps the BPD is quite high or getting quite dry in mid to late January, then the dehiscence or anthesis is elicited. So I want you to comment, if you would, please, on whether or not my understanding is correct. Is there a role to play in flower induction? Are there differences in year-to-year variation in the timing of flower induction, or is it primarily in that anthesis timing that is important?
Yeah, it's definitely all of the above, right? So we have the development of these pollen cones, and that I'm afraid I don't have any data on the development of them. I think that would be really useful here for teasing it apart, but 2 years wasn't enough time to do everything. And so I kind of see this as a series of processes, kind of like this Rube Goldberg device of this happens and then this happens. And some of these, the ones that I've been focused on are the kind of general ripening and then the processes of pollen cone opening and release. But the development of them, I think the fact that soil moisture in that spring is one of the best predictors for the differences in timing implies that it does have a lot to do with it. And I wish I had a plant physiologist to collaborate with on some of this stuff.
Do you have a sense for how well your models work or might work with other trees?
Yeah, there's a ton of variation. And something like oak trees, where when I showed that example of national stuff at the end, it works great for oak trees because they tend to release pollen at pretty similar times of year. Some types of trees are just way harder to predict than others. When there's a lot more variability between individuals, that can really make things challenging. And so I think what we're going to end up doing is, well, at least what I'm going to end up doing is focusing on the most important and easiest species and genera first, and then eventually tackling these tricky ones. And some of these things are going to be predictable, but others could be due to genetic variation, things which are very hard to guess about from first principles. So great for some things, mediocre for others. It's going to be an awful lot better than the commercial pollen forecast, so I'll tell you that.
Sorry, I was going to ask, if you hadn't brought it up, I was going to ask about the commercial pollen forecast. If they don't use the data from these like collecting sites all around, like what are they basing it on? So they're not using your models, obviously. Like, where's that coming from?
Well, there are a bunch of different sources of information. The word on the street is that, at least for some of these, I can't name names, but some of them use things like sales of allergy medication to get a sense of the timing of this. Some of them have access to some real data. There are a few pollen monitoring stations which post their information directly online without having to, and don't require it's just generally available. So there are a couple of the pollen forecasting companies that are trying to do some of the science behind this, but and I'm afraid I can't say too much more about that other than there's, I think, currently just no accountability. And so there's not much incentive for them to make sure that they're doing things right. Hopefully when we get this thing up and running and have a website that says like, oh, well, this company's model predicts 12% of the variation, like maybe then they'll have a little more incentive to do it right.
And a quick question. Clearly, when you're looking at pollen with these trees, you're looking at it as a disservice, but you talked about shade as being hopefully a ecosystem service. I guess my question to you is, given the kind of work that you do, have you been caught in a situation where you're being asked, what kind of tree should we be planting in this municipality? And if that is kind of a tricky kind of place to be, yes, you could put this tree, but because it doesn't produce pollen, but it might not have other. I'm just wondering, how are you navigating this? Kind of seems to me that you've got a tug of war between service to service when it comes to these trees.
Yeah, absolutely. Fantastic question. And that's just how it goes, right? There are our interactions with these trees are so multifaceted. There's 100 different ways in which you interact with the tree outside of your house, even if you aren't really thinking about it consciously most of the time. And some of these things are kind of annoying or somewhat dangerous, like pollen or like a tree falling on your house. But a lot of them are really good, especially in terms of heat reduction. The psychological benefits, the economic benefits are generally massive. And so what I think we need to do is to understand each of these different dimensions of it. What are the good things that a tree would provide, and what are the bad things? I don't think we should just ignore the bad things just because somebody likes trees as much as I do. Like, no, we should weigh all of these things together. And one of the pieces that's really been missing in a lot of this literature is understanding the spatial dimension. So like, what are the distances over which these effects are going to be felt? And so I think the thing to do is ultimately to combine all of these together and provide folks like urban foresters and other people planting trees. I mean, there's 1 billion trees planted each year in the US. So this is an important question. Let's give people the information they need. And I don't know, maybe by the time I'm like, maybe by the end of my career, we'll have some tools which can do a better job of saying like, hey, if you have this spot here, what are the specific ecosystem services and disservices that you would get from planting this species of oak here versus this species of maple or whatnot, and to understand it, perhaps unified through an economic valuation framework.
Great.
Okay, so there's another question.
Just a quick one, yeah. Is your understanding of from that once they open, they release all of the pollen, or like do they release on clothes?
Yeah, it's so what ends up happening is way more complicated than I had hoped for, and it is so there's. Let me see if I have. I just want to, yeah, let's go here. And so you can see all of these individual pollen sacs. And so so there's both these kind of scales opening and closing. And then there are these individual pollen sacs inside of it. Once a pollen sac opens, it's opened. However, it's often like when you're looking at a particular pollen cone or thousands of them, you see that often there are some pollen sacs open on a cone, but not all of them. And so I'm trying to think of something clever to do to tease apart some of the processes based on that information, but still considering it.
And part of why I was asking was that chart we were looking at where we saw the spikes and then kind of pause, and then another spike is, I thought like, oh, could it just be related to like the wind on that day? You know, like why did these scenes? Anyway.
Yeah. Wind plays a large role. I can show you quantitatively that yes, there's an effect. However, it is much more than just wind. Thank you all. Really appreciate your time.
This has been a production of Cornell University, on the web at cornell.edu.